Melissa George,
And I'm Fayza Mehrin.
and our study focused on evaluating the rate of microplastic filtration on Mytilus edulis, also commonly known as the blue mussel.
Microplastics pose a threat to our ecosystem by ending up in large bodies of water and being consumed by the organisms that inhabit these aquatic biomes. However, a species of mussels called Mytilus edulis are filter-feeding organisms that filter out water without any harm inflicted on them. The goal of the study is to assess the patterns in the filtration rate of the Mytilus edulis on different quantities of microplastics, which can serve as a model for scientists on developing environmental friendly solutions on microplastic contamination.
The question our study centered around was, what is the rate of filtration for Mytilus edulis? In order to reliably conduct the experiment and calculate the rate of filtration, our independent variable was the amount of microplastic, and the dependent variable was how long it took the group of mussels to filter out the microplastics. We hypothesized that the rate of filtration will remain the same given the controlled environment that the mussels were in and the eutropic qualities of Mytilus edulis.
In the study, two 2-gallon tanks were used along with an air pump, 2 air stones, sand and gravel which served as substrate, reverse osmosis water, sea salt mix, and a red plastic cup.
The procedure for this experiment was a critical 3-step process. Step 1 involved setting up the tanks. The tanks were first thoroughly cleaned to prevent contamination for the mussels, set up on a level location away from windows and ventilations to avoid external factors interfering with the experiment, and then they were filled with substrate about 3 to 5 inches deep in each tank. Step 2 consisted of mixing the salt water together. At the ratio of half a cup of salt per gallon of water, 3 gallons of salt water were mixed until all salt was dissolved and the water was clear. The water was then aerated and left alone for 2 days to stabilize and then was added into each of the tanks. Step 3 entailed making the microplastics. A red cup was cut into pieces less than 5 millimeters in length and were verified using a ruler. The pieces were then tested in a cup of water to make sure that they would either suspend or sink. Once the tanks were set up, the mussels were bought from a supermarket. The mussels were gently washed in running water to ease them from a freezing temperature that they were bought in to a somewhat room temperature. 5 mussels were then placed in each tank. The mussels were placed face down with the hinge side in contact with the sand to prevent possible smothering. It took the mussels about 2 days to acclimate and reposition themselves. Microplastics and powdered algae pellets were then introduced into each tank, and the experiment was run over 2 weeks, and the time was recorded to the nearest quarter mark when collecting the data. We were able to run the experiment with 10 and 15 pieces of microplastics, respectively. As you can see from the table, the time it took the mussels to filter out the 15 pieces was higher than the 10 pieces. We unfortunately could not collect data for the experimental group with 20 pieces of microplastics, as during the trials, 2 mussels in each tank suddenly died. Based on the data collected and calculated, the rate of filtration had some variation. Generally, for the group of 5 mussels, the more microplastic that was added into the tank, the more time it took for them to filter out the microplastics. Overall, the rate of filtration per mussel was about 4.88 hours. The rate of filtration increased by approximately 1.6 times between 10 and 15 pieces of microplastics. Based on the filtration rates, it can be assumed that in each trial, each of the 5 mussels filtered 1 to 3 pieces of microplastics.
The hypothesis for the study was not supported, given the fluctuations in the mussel's filtration rates. The study had 2 trials per experimental group, and in the process, some of the mussels died. Some factors that we infer to have caused this may be stress on the mussels, the rise in the levels of ammonia, and the lack of oxygen. This leads to our data to be inconclusive. However, The findings in this study display potential in the use of Mytilus Edulis in the development and solutions for microplastic contamination. As stated previously, the death of the mussels created a mid-study issue since the organisms released ammonia after their death. This then contaminated the water and killed off the other mussels as well. Additionally, the conditions that these mussels were in and the adjustments that they had to make to their new environment caused additional stress on the organism, which may have impacted them psychologically. For further research, some considerations are to change the tank environment and include live bacteria, as well as include more trials and decrease the quantity of mussels for the experimental groups. Regardless of the limitations that our study faced, the data accumulated and its following findings display promise for use of Mytilus Edulis as part of a solution to combat the escalating issue of microplastic pollution in marine and freshwater ecosystems. Thank you for your time.